Hello, my name is Alejandro Sánchez Alvarado. I am an investigator with the Stowers Institute for Medical Research, as well as the Howard Hughes Medical Institute. And what I would like to share with you today in the next few slides is a way to make the argument as to why we need to expand the numbers of model systems in order for us to understand not just life, but also human biology and the diseases that afflict us. And so to do that, I'm going to tell you a little bit about this idea of the origin and diversity of animals. We all know that there's an immense diversity of organisms out there. We all know about the principles of evolution and how we are related to each other by very, very deep associations with the origins of particular genes and the like. But even though we know all this, we know that it's possible, we really don't know what is possible in nature as it is right now. And the reason for that is because the nature of biomedical research is not really allowing us to actually look. We have to actually do the work with the systems that are available to us in order to ask questions that we think are relevant to understanding our own life as human beings, as well as life in general as a phenomenon that happens on this planet. So I'm going to give you an example of what I mean by this. So we have studied very, very few species thus far of the immense diversity of organisms that are present in the globe. And the main organisms we've used, and that the NIH and other funding agencies have invested a huge amount of resources for us to understand and use as vessels to generate knowledge and understanding of biological processes, are represented right here. These are incredibly powerful model systems that have, ushered a really profound understanding of biological systems and biological processes, starting from humans and mice, frogs and chicks, and of course the zebrafish as the representatives of the vertebrate world. And they are all being used in large numbers of laboratories, not just in the US, but across the world, to ask the questions essentially of what is life, how does it arise, and what happens when it goes wrong. Can we understand those mechanisms? Part of the process in trying to understand life has also involved the utilization and understanding of invertebrate model systems, which I'm showing you right here, C. elegans and Drosophila, that are an incredibly set of organisms to ask genetic questions associated with complex biological processes. So what we've learned from these animals is immense, or at least we feel that way because we have now reached a level of understanding of certain biological processes that our predecessors would just envy and would not be able to rival by any amount of experimentation at the time and observation at the time. But that's what we know. Those are the systems that we use. It's about 7 model systems in general, give or take a couple more. But it would be interesting to know and to get an appreciation of what is already possible out there by just simply studying nature, by doing what I refer to as inductive inquiry, which is not necessarily what we do in the lab today, which is mostly hypothesis-driven research. An inductive inquiry essentially gets called fishing expeditions and a few other monikers that don't make it very exciting and of course don't make it very fundable because it's exploration, it's curiosity-driven perhaps, but it's essentially not being buttressed by some hypothesis that will allow you to explore this diversity in nature. So I'm going to start by giving an example, which are protists. These are unicellular organisms, okay, that are no near similar to us, unicellular. And then the one that's most famous, of course, is Tetrahymena, which is about 40 to 48 microns in diameter. Okay? These organisms were selected for investigation many years ago because they display some really interesting biological attributes that made them vulnerable to experimentation and for us to understand major principles of life. And so from a single protist organism, okay, we were able to extract the following information. Ribozymes were discovered in protists. This actually resulted in a Nobel Prize in 1989. Telomeres by Grider, Blackburn, Shostak, and others also resulted in a Nobel Prize. A single unicellular organism, okay, producing 2 Nobel Prizes already. And probably many more to come because the first molecular motor was discovered in Tetrahymena as well by Gibbons and Rowe in 1965. And then, of course, histomodifications and epigenetics, the first demonstration this was happening by Alice in 1979 also took place in these organisms right here. What is interesting, though, just to put it in contrast, is that you'd think that this is really esoteric science, right? But all of this research was actually funded initially by the NIH, the National Institute of General Medical Sciences. Every single one of these bold proposals to do inductive inquiry in an organism that doesn't have hair, is not a mammal, in fact is a single protist, resulted in funding that provide us with tremendous understanding of biological first principles, okay? So what that suggests to me, essentially, is that things have changed a little bit, because if you were to come up with a single organism like this and propose to investigate this research, the likelihood of success right now would be pretty low. I mean, that's been the experience of many of my colleagues, primarily because we think that we have enough model systems available to us to ask all the questions we need to ask. Okay, fair. We can assume that that is the case. But what if I were to tell you that this is what we think protists look like, right? We know what is possible in the protist world. There's small things, microscopic, ranging in size from 40 to 48 microns. And what if I show you this other organism, which is about 20 centimeters in size, and is also a protist? It's a Syringamina fragilissima. So the phenotypic genotypic space that a single cell organism can occupy goes essentially 25,000-fold difference in scale. If I didn't tell you this was a protist, a unicellular organism, you wouldn't know it. And so this space that's being occupied by the single genome of the single organisms is huge. So what is possible in nature, we don't appreciate because we're not looking. Now, it would be fair if I told you this protist, this Syringamina fragilissima, was discovered recently. This thing was discovered in the late 1800s off the coast of Scotland, okay? And we don't know anything about it today. And my guess is that if Tetrahymena had all these secrets to teach us about, telomeres, ribozymes, molecular motors, what other secrets is this organism holding that we should really learn about? And we don't know. We don't know because we're not looking, all right? So if you now take a more objective approach to what this means, and you now look at the Metazoan tree of life, which I'm listing right here, okay, what you see essentially is that the 7 model systems that we utilize, these 5 vertebrates, okay, essentially are vertebrates. All these other groups are not represented with the same degree of abundance in the biomedical milieu, okay? If you look at the other 2 workhorses of genetics in the invertebrate world, and I would venture to say in all of animal genetics, C. elegans and Drosophila, we're talking again about 2 phyla, the nematodes and the arthropods, and these other phyla are not being studied. If you now count how many phyla make up the phylogenetic tree, which is essentially rooted in a single cohort of organisms down here, we're talking about 29 different phyla. Now I ask you, okay, what is the statistical probability that 7 organisms, such as these, will encompass the immense diversity of biological activities that are present in the Metazoan tree of life? Just do the math. That's 9 times 10 to the 5% of all animal species are represented by these 7 organisms. Is that statistically significant? Of course it's not. So what is the likelihood that every single biological problem that we really want to understand is going to be represented in these animals? Very low. That's the bottom line, it's very low. And if you were to tell me that it's not possible to do investigation in all these other animals because they're different, they're weird, they're hard to access and so forth and so on, that's a good argument 20, 50, 100 years ago. But not with the technologies that we have accessible to us today, where we can actually bring science to nature as opposed to the other way around, where we have to bring nature, domesticate it, and hammer it in the lab. I think we can do the opposite, and we should be able to do that nowadays. So why do I say that? Because there's a cohort of phyla for which we know so little relative to what we know for the 7 organisms. So the likelihood of us not making equivalent discoveries and significant and insightful in the next 10 years, if we were to dedicate ourselves to study these animals in relation to what we have studied, is 0. This is a waiting. This is a mine of new discoveries waiting to be picked by people willing to actually do inductive inquiry, buttressed, of course, by the scientific method, right? So normally, the argument is as follows. We have this really high resolution of established model systems. We have genomes. We have ways to edit their genomes. We have genetics. We have tools. We can modify, see every single nucleotide in the genome, and that's great. That's the high resolution of established model systems, but you're essentially limiting yourself to the biological attributes represented in that very small cohort of animals. Whereas the low resolution of emerging model systems is seen as a deficiency because, well, they're hard to bring to the lab, they don't have all the tools, maybe the genomes are not ready, and so forth and so on. Well, I liken this rationale as saying that the high resolution of established model systems, that's like mapping already discovered continents. We know more or less what the biological activities are going to be in these systems that we have studied for so long and in such great detail in the present time. Whereas working with these other systems allows us to discover new biology and therefore discover new continents. And that is the value of being able to bring other model systems into the milieu of biomedical research, right? So I think there is merit to try to develop the necessary granularity and tools to exploit all this new biology that we're not even aware is probably happening right there presently as we look out the window. We don't look. We're not encouraged to look, all right? We're encouraged to do other things. And for good reasons, obviously, but we're encouraged to do other things. And I think there should be room for both. Okay, so what I like to do is to give you an example of why looking at weird animals might actually be a perfectly viable and useful proposition to understand, our own biology. Okay, so I'm going to give you a short story of discovery that was done by looking at the excretory system of an animal that doesn't look anything like us, which are these Planarians that I'm going to tell you about in just a few minutes. And what I would like to do is to walk you through what we think we understand about the evolution of the excretory system, the kidneys, and the corresponding or attending diseases when things go bad. Okay? So what do we know about the kidney? Well, a lot has been learned in the past 30 years. But for some reason, the kidney has not really attracted as much attention as, say, the heart or the brain. And it's really interesting when you start looking at the old literature. And by old, I mean just 1960s. I don't mean like early 1900s, just 1960s. So for example, we know that if you take a urine sample of a human at any given time, there's a huge amount of daily shedding of renal tubule epithelial cells. If this is constantly happening and you're not replacing it, we will be dead. Our excretory system will be completely spent. So that means that that tissue is being reconstructed on a daily, if not hourly basis to allow us to keep that system functioning for the 80 years or so that we might actually live for, if we're lucky enough. Secondly, we know that there is compensatory renal growth following nephrectomy. And this has been discovered in a report in 1975. If one kidney is removed for a transplantation, the other kidney will grow to compensate for the function of the missing kidney. So if those tissues were really static and there was no growth, this would not be possible. So we already know it's happening. We just don't know how, but it's happening. Third, we know that in pediatric cases, where either a solitary or a single functioning kidney exists, we also see renal growth. Suggesting again that these tissues can add cells and can add size to the structures as well. So it's not a static system, it's a dynamic system that is constantly undergoing change. So great. So where is that change coming from? Is it that you have stem cells residing in there, and if they're there, why can't we regenerate these things? So the most recent discovery was published in 2014 by Rinkovich in Irv Weissman's lab, showing that by doing clonal analysis in mice, there's constant repair and growth of the renal tubules. They were able to identify by clonal mapping individual cells that are dividing, producing new cells, and replacing those cells that you're shedding as we urinate on a regular basis, suggesting that the tissues are really constantly rebuilding themselves. Okay, so that means essentially that the kidney really is a dynamic system, developmentally speaking, okay? So then what do we know about the evolution of kidneys? Right? So if you look at the papers that are cited in the literature about the evolution of kidneys, you always see a reference for this paper from Howard Smith, 1943. And if you look at the evolution of this tree of life, what you see essentially is that this is only the lineage that gives rise to us. This is the so-called deuterostomes. Most of the non-deuterostome invertebrates are absent from this tree. Why? Because we're assuming that our kidneys are so different from everybody else's that we invented kidneys. The deuterostome invented kidneys, and the other systems are doing something completely different, analogous but not homologous to our excretory system. And so it stops right there, somewhere down here, showing that what the structure of the nephridia or the excretory tubules are in this system. And there is good reason to, believe that that might be the case. And all you have to do is just look at diagrams of the kidneys we're going to show you. But before I do that, what I would like to do is work from the following hypothesis. And we hypothesize that the kidney's ability for growth and repair, rather than being of vertebrate origin, is evolutionarily conserved and dates back to the evolution of the excretory system itself. And I'm hoping I'm going to be able to provide you with sufficient evidence to support this hypothesis. So why do we think that the kidney was invented by vertebrates? Well, you have to look at the unit, at the functional unit of the kidney. In this case, the nephron, okay? So what you see essentially is that here's where the circulation goes. It goes through this filtration apparatus, referred as podocytes, and it goes into the proximal tubule, the distal tubule, urine is made, and it gets excreted. And that's what that structure looks like. Okay, great. We need an inverted model system for gene discovery. How does Drosophila do it? Well, there's no separation between the circulatory system and the excretory system. So they're not really homologous. They're sort of analogous. And so our nephron is way more complex than the flies. So there's only a few things we'll be able to study in the flies. Okay, what about C. elegans? C. elegans must have an excretory system. Yes, they do. But it's a single cell. A single cell, right? So if you look at this evidence, you say, oh, all invertebrates, none of their excretory system looks anything like ours, then you say, oh, great, vertebrates invented the kidney, right? And so you look at the ancestor of the entire tree of life, let's say cnidarians, okay? Well, they don't have an excretory system. So that lends to the idea that the excretory system was invented recently in evolution, and you can build your arguments and say, vertebrates are the way to go, and so therefore we need to only study the kidney in the vertebrates, and the other organisms might provide us with some insight, but not necessarily the insight required to dissect diseases, functions, and malfunctions, and the like. But if you look now at a close relative of the vertebrates, in this case Amphioxus, okay? What you see here is that, yes, you have a circulatory system, you have this filtration system, and these cells that have a cilia that beat to make the fluid flow into the tubule so it can be urinated. Now, this group of animals, which is highly understudied and contains the largest collections of body plants in the planets, and for which we know almost nothing, referred to as the Lophotrochozoans, have very interesting excretory systems. So you can see a Planarian, which is the one I want to tell you about. That structure is very similar to the Amphioxus in that it has a filtration apparatus. It has cilia that allows the flow to go into these tubules and then be excreted. So we ask the question, how similar is this system, excretory system in Planarians, to the system that is found in the deuterostomes? Do they share a common ancestor or not? Or do they evolve independently? So here's what the excretory system looks in Planarians. There is no singular kidney associating one organ. The excretory system is distributed throughout the body of the animal. And you can see all these little white dots right there. Those are those cilia that beat, so-called flame cells, that actually move the fluid that is going to be excreted into the tubules for excretion in the animal. They are distributed throughout the body of the animal. You can see the head right here and the tail of the animal. These have different shapes or distributions along this tissue. And what is nice about this tissue is that not only does it have a proper distribution along the anterior-posterior axis, but also along the dorsoventral axis. And you can see here a depth pseudo-coloring of where the flame cells, the cells with cilia, and the tubules are in this organism. We can visualize these things. All right. So we refer to these structures as protonephridia. I mean, that's what the literature refers to them. And it's part of the excretory system of these organisms. So this is what they look like. So we can now begin to visualize them in 3 dimensions. And what we see is that the fluid will enter through here, go through these proximal tubules, empty into these distal tubules, and then these distal tubules enter into the exit tubule where the excretion occurs in these animals. Okay? So it's a complex system. We can visualize it. If we perturb it, we should be able to see a specific perturbation. So the first thing that we did, particularly a graduate student in my lab, Han Vu, was to essentially clone all of the genes that are known to be solid carriers in this animal. And to do that, we look at all the solid carrier proteins that are found in mammals, and we clone all the homologs in Planarians, and then ask, are they expressed in the Protonephridia or not? And it turns out that they are. But they're not only expressed in a particular, in a random location, they're actually expressed in very precise locations along the proximal, distal, and collecting duct of both the Planarians and homologous to the situation in mammals. Suggesting then that it's not just gene expression, but it's structural along the length of the tubule, providing the tubule specific identities and functions along the route by which this fluid is going to be excreted. So solids can be brought in, others can be emptied out. So we tested that by looking at the genes associated with controlling pH, carbonic anhydrases, and these solid carriers. So we looked and found the homologs in both mice and vertebrates, and Planarians. And what we see essentially is that when we perturb these genes by RNAi, we use a pH-sensitive dye, we see that essentially the pH in the animal changes. And these genes are located in an anatomical position that is homologous to the anatomical position of the mammalian nephrons. That's millions of years of evolution in a non-deuterostome invertebrate. Coincidence? Yes, it could be coincidence. So we thought, how much of a coincidence is it? Can we actually go further? So the function tells us already that it's functionally conserved. So now we can ask another question, which is, do these homologies that we begin to find between the nephrons and the protonephridia extend to common pathologies? Because if the diseases are conserved molecularly, then it's no longer really a coincidence, right? So we ask the following question. We know that in the mammalian kidney, mutations on these two molecules, NPHS1 and NEPH1, cause what's called slit diaphragm loss, which results in massive edema in humans. Those kidneys look awful. So here's an electron micrograph of what I mean. So here's the cell that makes the filtration. This region right here referred to as podocytes, and you can see those right here. This is where the fluids are being filtered from the circulation back into the excretory system. We now take electron micrographs of our Planarians in the same location of the excretory system, the so-called flame cells. And what we noticed was that, yes, there is cilia in there, which is absent here, but we saw these beads along the edge of the animal that resemble significantly the structure of these podocytes. Turns out that when we do in situ hybridizations of these molecules, that's exactly what they're expressed, which is exactly what they're expressed in the vertebrate condition, right? So what does that mean? It means that if we now perturb it, we should expect to see edemas, right? So we go ahead and silence those genes by RNAi, and that's exactly what we get. Planarians now cannot excrete, and they get edematous, and eventually begin to blow up and then just die. If we take an electron micrograph of these animals after RNAi, this is what we find. We find that those podocytes, which is here in the normal condition, you can see those really nice filtration sites, it's gone in both of these. And this is indistinguishable from the vertebrate condition. When those genes are absent, the podocytes are not made, edema occurs, and there's renal malfunction. So that's already another line of evidence to suggest that it's not just that the genes are expressed in the right location and that the structures are homologous, but the pathologies are also conserved. So we can go one step further and look at other pathologies that are found in the kidney for which there are no invertebrate model systems for us to compare. So the cystic kidney disease, or diseases, so to speak, they can only be modeled in mammals so far. And so we've asked the question, well, can we find molecules associated with cystic kidney disease in the Planarian genome, and what do they do in Planarians? So things like the nephrocystins, shown right here, which actually cause diseases like nephronephritis. And when we look at the genome, what we find is that we find a lot of the genes that are known in humans to be defective to cause these diseases, and interestingly enough, these genes are missing in Drosophila and C. elegans. So that's probably why we cannot model those diseases in these organisms, because, well, they're lacking the genes. But Planarians have those genes. So what does that mean? It means that Drosophila and C. elegans lost those genes. Planarians have them, humans have them, the ancestor should have had them both, because they couldn't have invented both of these genes independently, suggesting that the origin of the excretory system is really conserved. They share a common ancestor. So now we can ask the question, if that is true, and we go ahead and perturb the function of these molecules, will we get cystic kidney disease or not in Planarians? So that's what we did. And so we can see here in the nephrostatin 8, what you can see essentially is the solute carrier in purple is much more, in red, is much more abundant than in the control right here. And when you superpose with the tubules, you can see that you actually have cysts in there. This is very similar to the situation that you see in mammals. Okay? You can now reconstruct this whole image three-dimensionally, and you can now clearly see where the cysts are along the protonephridia of these organisms, suggesting then that it's not just that the molecules are shared, also the pathologies are shared. We can take this even further and ask the question that is associated with cystic kidney disease, which is that the kidney somehow senses the rate of flow. And when that is perturbed, that also causes cystic kidney disease in humans. So now we can ask the question, if we perturb flow in Planarians, in the protonephridia, will we get cystic kidney-like disease in Planarians or not? So we do that. So we look at the cilia. Here's a control. The cilia are beating just fine. We get rid of a molecule that's associated with the flagellar motility. And you can see that the cilia are not beating normally. So this will actually affect the flow rate of the organism. And you can look at the other molecule that's involved in making the cilia, LRRC50, and there's no movement either. And when we do that, just by perturbing the flow, by targeting these molecules with RNAi, we actually were able to produce cysts or cyst-like structures in Planarians. So it's not just the genes, but it's also the function, the sensing of the flow, suggesting then that this is way too much of a coincidence, and that in fact, the ancestor to the vertebrate kidney was shared by both the protostomes and the deuterostomes. That then provides us with a novel system for us to actually do gene discovery. We don't know why sensing of flow malfunction results in cysts in mammals. We just don't know why that is. It's difficult to do those experiments in mammals for very good reasons. But if you have a model that actually shares structurally, it shares genes, and it shares malfunctions, that is as good a model as you're possibly going to get to be able to do gene discovery and try to identify the root cause of some of these diseases that occur in mammals. So we think that it's not just the function and the structural conservation, but also the fact that you can get cystic tubules by just perturbing flow. And that disease arises from the misregulation and misproliferation of the stem cells that probably reside in the tubules to allow for this overgrowth to take place. And so I think that this system will allow us now to uncover the molecular and perhaps cellular basis of the early ontogenetic processes that result in the production of some of these diseases, which are very, very difficult to sort out in mammals because it's expensive to do these experiments. So I hope that I have convinced you that there is benefit in looking at organisms that are not necessarily similar to us. Because I think that if you make the argument that an organism that has a particular biological attribute is weird, but it's, oh yeah, that only happens in Planarians, or that only happens in protists, okay? But it's not a mammal. When you make that argument, you're essentially denying evolution. It's the equivalent of being a climate change denier. Because you're saying that because it only happens in Planarians, it's only specific to Planarians, it's only specific to those organisms, when that is not the case. That is the power of evolution in trying to help us understand the ontogeny and the development of these diseases across the phylogenetic tree of life. And all of these organisms will have specific attributes that are exaggerated that we should be able to exploit to be able to inform our own biology. So I think we need more of these types of paradigms. So we think that the bilaterian secretory system shares a common ancestor. We think that these complex topological and molecular similarities are inescapable. They are there, they are verifiable, and therefore suggest some conservation. We also think that the excretory system genes that are shared with vertebrates clearly have been lost in Drosophila and C. elegans. So yet a need for identifying new model systems to fill in those gaps. And then the shared pathologies, you know, the podocyte deficient edema and the CKDs really suggest that Planarians and perhaps other organisms like it will allow us to identify orthologous invertebrate paradigms in which to model vertebrate kidney development and disease. Thank you.